Hello lovelies, in this video the brilliant Dr. Upwards is going to be talking about control of blood glucose. Now this is one of the topics, there are lots of topics in A-level biology, where your handwriting can let you down. Because if you're not very clear to the examiner, if the examiner cannot read what you're talking about, glucose, glycogen, glucagon, then they cannot give you the mark. So please, this is one of the topics where your handwriting and your spelling is really important.
Okay, so we're going to look at controlling blood glucose concentration as one of our obviously examples of controlling a factor in the body using the endocrine system. So we mentioned this previously, but just to recap why it's important that we need to make sure we maintain our blood glucose levels. So it's just stating about 4 to 8 millimoles per dm cubed of blood. That's the ideal range. If it's below 4 or above 8 on these little prick machines that you can get where you can test your blood, where you do a finger prick, put a little bit of blood onto a little strip and the machine will read blood glucose content. They are obviously got better other equipment now where you can use kind of a phone that it talks to a little patch that you have that sends it to an app on your phone, or you can scan the little patch with your phone and it will tell you your reading as well. People who have type 1 diabetes are very familiar with these different technologies. So why is it important that we keep our blood glucose level within this range? Well, if it drops too low, then cells might not have enough glucose for respiration and they may not be able to function normally. Brain cells are particularly sensitive to this. Your brain actually needs quite a lot of glucose to function, and it requires a lot of energy. So if your blood glucose levels drop, similar to if your blood oxygen levels drop, then your brain is very particularly affected. If blood glucose levels are too high, it can disrupt the water potential, like we said. So blood plasma, it can change the water potential and then have osmotic effects. So cause water to start moving in and out of cells if the blood plasma and the tissue fluid aren't isotonic to your cell's cytoplasm. So we don't want to get it too high for that reason. So let's have a think about how we're going to control this then. So the blood glucose concentration is going to be controlled by the parts of the endocrine system. So it's to do with hormones, and we're using glands to be able to control this. So the receptors that are going to be detecting the blood glucose levels are located in regions called the islets of Langerhans, which is the endocrine tissue inside the pancreas. We'll look at that in a bit more detail in a second. The signals that we're sending is hormones. So hopefully from GCSE, we should remember that hormones are insulin and glucagon, and they are both secreted by the pancreas and they'll be traveling around in the bloodstream. And then the effectors which are going to cause our blood glucose level to either increase or decrease are going to be in multiple organs and cells. So the liver, the muscle cells and fat cells are the target cells of these hormones. Target cells means they have specific receptors on their cell membranes for insulin and glucagon. So that only cells that have these receptors are going to respond to insulin and glucagon because they're the only ones that have the receptors that will bind to them. So they act as effectors and they're going to help us either raise or lower our blood glucose levels. Okay, so the main cause of blood glucose increasing is when we eat. So when we consume carbohydrates that are then broken down into monosaccharides, including glucose, and they're absorbed into the blood through the small intestine. So anytime you eat after food, you're going to increase that blood glucose level when it's absorbed into the blood through the small intestine walls. There are some other ways that your body can increase your blood glucose level. So glycogen, remember, that is the storage molecule, a long-chain carbohydrate that we use to store glucose in the body in animals. So glycogen stores in the liver are broken down to release glucose. And this process is called glycogenolysis. It makes sense. It's basically glycogen-olysis. So if we think about what we know, so we're talking about breaking down glycogen in the same way if we think about hydrolysis. What lysis means is to split. So we are basically splitting glycogen. The other thing the liver can do is that it can actually convert other substances into glucose. So we can take lactate, which is obviously produced from lactic acid, which is made in the muscles during anaerobic respiration and finds its way into the blood, and it can find its way to the liver. It can use lactate and convert that into glucose. amino acids, for example, pyruvate, which is a protein that also is going to be produced and going to be able to be released into the blood from muscles as well during respiration. So that can be used as well, other amino acids as well. And then glycerol from fatty acids from fat stores can also be released, sent to the liver, and we use these to create glucose. And if you think about it again, this is another term that we need to know. There's lots of words in this topic that we're going to have to get used to, and they all sound very similar. But in this case, we've got gluconeogenesis. So gluco meaning glucose in this case, neo meaning new, and genesis meaning to make or the start of something. Thinking about the book of the Bible, for example, the book of Genesis, that's about the creation of the earth. We're making something new. So glucose, neo, new, genesis, making new glucose. The liver can also do it. And in both of these cases, whether we are converting from glycogen to glucose or we are creating glucose from other molecules, we're then going to be able to release that glucose into the bloodstream, and therefore blood glucose levels are going to increase. Blood glucose levels can also obviously decrease for various reasons. So when the liver removes glucose from the blood and converts it to glycogen, so this process that we looked at initially is reversible. Hopefully we remember this from GCSE, because this is how we should remember that glucose is removed from the blood when insulin is released from the pancreas in order to lower blood glucose levels. So that's one way which we should be fairly familiar with. The other thing to think about is sort of the opposite of eating food, really. You eat the food and the carbohydrates, and that increases your blood glucose level. And then the opposite to that is if you do some exercise. So if you need to use up that glucose during respiration, then you're going to reduce your blood glucose levels, because glucose is going to be being removed from the blood and going into the muscle cells. So one more thing, which is kind of an extra thing, but it's good to know. So drinking alcohol can actually prevent the liver from producing glucose. It slows down the liver's function because instead of dealing with the glucose, it's helping to sort out the alcohol in your system. So it can actually cause a decrease in blood sugar levels, which is good to know. Endocrine glands are the glands that secrete hormones directly into the blood. Well, exocrine glands secrete products, not just hormones, but other things as well, into ducts or tubes, vessels. The pancreas contains both exocrine and endocrine tissue. The exocrine tissue secretes digestive enzymes into the bile duct and the pancreatic duct. And the endocrine tissue, the islets of Langerhans, secretes hormones into the capillaries. So here you can see I've got my exocrine pancreas cells, which are situated around my islet of Langerhans. And these are secreting the digestive enzymes that go into the pancreatic duct, which then joins up with the bile duct. And so the enzymes from the pancreas and the bile from the gallbladder will together go into the duodenum, or the first part of the small intestine. So pancreatic amylase is one of the enzymes produced by these cells. This digests amylose into maltose. Trypsinogen is another enzyme. It's actually an inactive protease enzyme, so it breaks down proteins. And when it gets into the duodenum, it will form trypsin, which is the active form. And the exocrine tissue also makes lipase, which are enzymes that digest lipids. So yeah, these are secreted into the main duct, which is here, this kind of brown one, and you can see it joins up with the bile duct here, so they join, and then they go into the first part of the duodenum, which is the first part of the small intestine. The pancreatic islets of Langerhans are the endocrine tissue in the pancreas, and we've got two types of cells in the endocrine tissue. We've got the alpha cell and the beta cell. The alpha cell secretes glucagon into the bloodstream, and that's obviously a hormone, and this raises blood glucose levels. The beta cells secrete insulin into the blood in order to decrease blood glucose levels. So you can see I've got these little arrows which are showing these yellow particles of insulin being released by the beta cells and then going into that capillary, and then all of those capillaries you can see coming, the blood vessels will come out of the pancreas and then they will go in directly into larger blood vessels, and then that will transport around the body, which is how the hormone will get to its target cells. As with all of the other organs, we need to know our pancreas histology, so we need to be able to recognise these structures in actual stained pieces of tissue. So here is some stained piece of tissue for the pancreas underneath the light microscope. So you can see kind of, I've got 3 islets of Langerhans here. I've labelled one of them for you. And you can see obviously those cells are like sort of that circle, large circle group, and they look very different to the exocrine tissue. The white, thick kind of white gaps you can see is the connective tissue that forms between the lobules. So the lobules are the groups of the exocrine tissue. So here you can see one lobule that I've kind of circled around. So you can see it's got the connective tissue going all the way around it, and we've got that one lobule there. So these groups of exocrine cells are known as acini, and if you just have one group of them, then they are an acinus. But if you have multiple of them, then you can call them acini in plural. So at the centre of these acini, then they have tubules, and these drain down into the pancreatic duct. So they're secreting these enzymes constantly into these tubules. The tubules then drain down into the larger pancreatic duct, as we said, joins up to the gallbladder, the bile duct, and then that all goes to the duodenum together. And then what you can see here is that blue kind of colour, that is a capillary. And you can see here that obviously the islets of Langerhans are kind of arranged around that capillary. So they will be secreting their insulin and glucagon straight into that capillary, which will then flow down and flow out of the pancreas into the bloodstream. We need to know how beta cells actually control insulin secretion. Beta cells have both potassium and calcium ion channels in their cell membrane. At normal blood glucose levels, the potassium channels are open and the calcium channels are closed. The beta cell, we say the membrane is polarized because there is a difference in charge. It is more negative inside and more positive outside. When blood glucose levels increase, more glucose enters the beta cell by facilitated diffusion. More glucose increases the rate of respiration, so more ATP is produced by the mitochondria. Potassium channels close, so potassium ions build up inside the cell as they cannot leave. This depolarizes the membrane by making the inside of the cell more positive than the outside. The depolarization of the cell causes the calcium ion channels to open. Calcium ions diffuse into the beta cell and cause vesicles containing insulin to move and fuse with the cell membrane. This causes insulin to be released from the cell through exocytosis. The movement of vesicles and exocytosis requires ATP, which is present in large quantities in the cell now.
Right, so let's look at what actually happens when blood glucose is too high or too low. Now, this is mostly similar to what you would have done at GCSE, but there will be some differences. Mostly just a little bit more detail. So initially, obviously, the change is detected. So when blood glucose is too high, the change is detected by beta cells. And I've used a little Greek symbol beta here. So the change is detected by the beta cells because they start increasing how much glucose is moving into them by facilitated diffusion. So that's how they kind of detect this increase in blood glucose. So in response, this kind of increase in glucose in the cell causes vesicles that contain the insulin to move towards the cell membrane and then release that contents into the capillaries. Now, this is very obviously similar to what we previously looked at with the idea of an action potential, causing those voltage-gated calcium channels to open, causing those vesicles of neurotransmitters to move, fuse with the membrane, and release the neurotransmitter. This is very similar. So glucose concentration increasing in the cell causes these vesicles of insulin to move towards the cell membrane, fuse, and release it by exocytosis. So then the insulin is going to travel to our target cells. We already talked about the fact that they could be liver cells, fat cells, muscle cells. So they're going to travel to the target cells, and they're target cells because they have those receptors. So they're going to bind to the receptors on those cells. And what they cause is they cause them to take up more glucose from the blood. How do they do that? Well, what they actually do is they trigger the cell to increase the amount of glucose transporter proteins, so those transport proteins that is going to be doing facilitated diffusion of glucose. They increase how many of those are on their cell membrane. So if they have more of those in their cell membrane, they've increased their permeability to glucose, so they're going to start taking up more glucose from the blood. And that's how we get our blood glucose level to decrease. The liver takes this sort of one step further in the fact that we've also said about how the liver plays a role in controlling blood glucose levels. So as well as increasing its permeability to glucose, it also starts converting glucose into glycogen to store it. And that means that it's going to be sort of removing that glucose concentration from the cells because it's converting it into glycogen, which is an insoluble storage molecule. And so that means that it can maintain that concentration gradient. So it means that there's always a low concentration of glucose in the liver, higher concentration of glucose in the blood. So it maintains that concentration gradient and makes sure that glucose is moving into the liver cells. Okay, so that's what happens if blood glucose goes too high. What happens if blood glucose goes too low? Well, it's very similar at the start. The change is going to be detected, but this time by both alpha and beta cells. Now, this is important because we can't have glucagon and insulin released at the same time. They work antagonistically to each other, like muscles that we talked about. So they are not able to work at the same time because they counteract each other. So when blood glucose drops too low, the beta cells detect this because the concentration of glucose in their cells is going to lower. And so they stop releasing insulin. So those vesicles of insulin stop moving to the cell membrane and it stops releasing insulin. But the alpha cells detect it and they start releasing glucagon. Again, the glucagon travels in the blood and it binds to target cells on the liver. And so it binds to those receptors, the cell membrane of the liver. It's going to cause a couple of things to happen. Mostly, it's going to cause the liver to do the opposite of what happened when glucose is too high. So it starts breaking glycogen down into glucose, which, as we said before, is glycogenolysis. Remember glycogenolysis, the splitting of glycogen. So it's going to start doing that to release glucose into the blood. The glucagon also activates enzymes which are going to start the process of gluconeogenesis in the liver. Remember, that means making glucose from other molecules, so things like lactate and pyruvate from muscles and glycerol from fat tissue. So the liver's going to start making more glucose from those kind of products that we have from fat tissue and from respiration in muscle cells. All of this leads to more glucose being released into the blood, which raises those blood sugar levels. And so we go back towards the optimum. So that's the pattern. We need to be able to describe that and what happens for both of those, and pretty much in that level of detail, and be able to kind of think about how these messages and signals are getting along. So thinking about the receptors, shapes fitting together, the fact that those trigger a cascade, which we're going to look at in the next video. So how we have these idea of once the insulin binds to the liver, how does it actually trigger glycogenolysis or gluconeogenesis? Because the insulin itself isn't moving into those cells, it's just triggering other processes to happen in those cells, which is what we call secondary messengers, which carry that signal into the cell and cause that change. So we're going to have a look at that in the next video, but it's mostly thinking about, can we remember which cells do which, alpha and beta, which hormones, and what effect those hormones have, and be able to explain how that either raises or lowers the blood glucose levels.
Ouch! This is why in some videos I explain scratches.